Okay, so I'd like to take a moment to introduce our speaker this evening. So Robert Gish will be talking to us about primarily hepatitis B, but also touching on Hep D. So a little bit about Dr. Gish. He was actually in pharmacy school first and then went on to obtain his medical degree from the University of Kansas in Kansas City. He completed his internal medicine training at UC San Diego, and then his gastroenterology and hepatology fellowship at UCLA, including a transplant year. Dr. Gish is an accomplished physician clinically and academically. He is a fellow of the ASLD, the American Society of Transplantation, and the American College of Physicians. He has served on the editorial boards of multiple prestigious journals in GI and hepatology, and co-authored a public health policy for liver health in Vietnam, focusing on Hep B. He is currently assisting with the development of viral hepatitis healthcare policies in Georgia, Armenia, and the Philippines. Dr. Gish holds many clinical professorships. He serves as an adjunct professor of medicine at the University of Nevada School of Medicine, as well as in pharmacy at the SCAG School of Pharmacy in UC San Diego. He is also a clinical professor of medicine at Loma Linda, as well as staff physician at Loma Linda's liver transplant clinic in Las Vegas. He is currently providing care in person and through telemedicine at various clinics in San Diego, Folsom, San Jose, Santa Rosa, Valley Springs, and Las Vegas, Reno area. Welcome, Dr. Gish, and thanks so much for being with us here today.
Thank you, Stephanie. A great introduction. Welcome everybody to a hepatitis B evening. I'll be building in a little bit on Delta into this, but my proposal to the societies is to have a talk next year on Delta therapeutics, actually Delta epidemiology and natural history and therapeutics. That's really an evening into itself, especially because of the recent purchase of Mir Pharma by Gilead with a Delta therapy that looks like it'll be available in the US sometime next year or at the latest early 2023. It's already conditionally approved in 3 countries in Europe and being also approved in Russia and being used clinically. Let's talk just for a second. My disclosures are on my website. I collaborate with a large number of hepatitis B companies. My website's also a resource for you. These slides can be shared through your society or through my website. Please feel free to use those, share those out for everybody's edification. Just a little bit of warning on these slides. There's a lot of information on each slide. I'm not going to be reading through each. I'm just going to be bringing forth what I consider key information you need to know about drug development and the excitement that I see that's happening in hepatitis B, and that we will have a much higher functional cure rate than 10%, I think, in less than 5 years. And then within 10 years, we may even be closer to a complete cure or sterilizing cure in some patients. I'd like to start out with just a little bit of ground rules in terms of when we're talking about hepatitis B. So when you're evaluating a patient for hepatitis B, there's some really important terminology you need to think about. Of course, this triple panel is what you should be ordering for everybody. You probably see in charts people getting an anti-core IgM, which is nearly worthless, except in the clinical setting of somebody you think has had acute or has acute hepatitis B. So you often have to reorder that. I've been working with Quest and LabCorp to get the panels updated, so it defaults to what I call this chronic panel or assessment for chronic disease. Another piece of fake news that's been out there for a long time is that core antibody has a high false positive rate. This is not true. Its false positive rate is about 2 per 1,000. So when you see a core antibody test, that person has cccDNA in their liver. If it's core alone, that person has been exposed to hepatitis B. They may have cleared surface antigen, which we call a functional cure, which is a bit of an oxymoron, but it's something that we built just to give people a little bit of hope. And you have to really think about core antibody in terms of reactivation or evaluating for occult hepatitis B. These patients do not need vaccine boosting. Unfortunately, that's still in the hepatitis B ASLD guidelines for unknown reasons. There is no data that administering a vaccine to a core positive patient provides any clinical benefit. You might get a little positive or boost in the surface antibody, but that does not mean that that person is going to have any better immune control. So when you see surface antibody with core antibody, that means immune control. They still have hepatitis B in their liver. Even though it's incurable, we use this term functional cure, and you should not be using the term natural immunity, another oxymoron. So not curable. S loss is our short-term goal in terms of managing our hepatitis B patients. I also want to highlight that there is this new wave of terminology that when you say LFTs, you mean bilirubin, albumin, INR, you don't mean liver enzymes. You really need to separate out enzymes from function. That's both from a professional basis and also when you're working with patients to have them clear that their liver might be inflamed, but their function is normal if that's the case after you assess the bilirubin, et cetera. The guidelines, there's very complex guidelines out there. There's 20, 30, 40 pages, depending on which society you look at. And these guidelines are so complicated, they really serve as a barrier to treatment. Too many gray zone patients, according to the recent NIH database that was assessed as well. Where we're moving towards, we're not there today, but the guidelines are being simplified. There's a really nice, simple guidelines on the University of Washington hepatitis B website that I helped co-write. But I'm going to make a forward-looking statement that within 3 to 5 years that we're going to really be thinking about treating any patient who is DNA positive. It's going to look a lot more like hepatitis C, right? You get an antibody test, you have an RNA, RNA is positive, you treat. Can't get much simpler than that with just, obviously a few caveats. Now there's another move that's taking place in the hepatitis B world. There's actually two things happening in parallel. So you really need to be ready in the next 3 to 6 months to see a statement come out from the ACIP to vaccinate all adults for hepatitis B. Now we want to harmonize vaccinate all adults for test all adults. And Hepatitis B Foundation, where I'm the medical director, we have been recommending to test all adults for hepatitis B since 2013. But the CDC and the Preventative Health Services Task Force just hasn't caught up with that simple recommendation. But we think there will be a big shift because if you're going to vaccinate all adults, you really shouldn't be vaccinating people who have hepatitis B. You shouldn't be vaccinating people who are core positive. So the only way to know is to test all adults for this triple panel and only vaccinate people who are triple panel negative. If they're core positive alone, you're going to educate about reactivation, potentially assess for occult hepatitis B. And remember, treatment isn't just giving somebody a pill or an injection. Treatment is about infectivity, laboratory testing, screening. More importantly is surveillance. Remember, screening is not repeated testing. Screening is the first test. Surveillance is ongoing testing in your hepatitis B patients. Imaging, education, lifestyle management. And hepatitis B is a stigmatizing disease. There's a social stigma and there's something called self-stigma. So be culturally aware. And this is one of the issues. Yes, you're supposed to be taking a risk history, but taking a risk history has its own impact on patients and their care. So that are a couple of really important issues before we jump into new treatments. I'm going to spend a little bit of time on this slide because as I move through the rest of the presentation today, I'll be moving relatively quickly, highlighting key points for you. But this is a complex virus. The virus is taken up into the liver cell, first by binding a heat shock protein, which then binds to the virus to the NTCP receptor. And this entry inhibitor that just was purchased by Gilead from Mir Pharma is an entry inhibitor. It binds to that bile acid receptor, and it looks a lot like the surface antigen in terms of the structural protein. Now, this entry inhibitor doesn't have a lot of effect on hepatitis B, but it has a big effect on Delta. And we're expecting this to be approved as a monotherapy for chronic administration, hopefully sometime next year. It's already approved in 3 countries in Europe, conditional approval. But it eventually will be used in combination. We'll be back, actually, believe it or not, using interferon with this medicine called bulevirtide. And there will be other options as Eiger, a Bay Area company that has lonafarnib and lambda interferon in a large global phase 3 study that also would be more finite therapy for many patients. Finite meaning with lonafarnib and with lambda interferon, you'll be treating for 6 to 12 months and then seeing who has a maintained virologic response, which could be up in the 40% range. And then you may retreat those patients or treat them with bulevirtide. So there's gonna be a lot going on in Delta. So another part of these base rules for hepatitis B is any patient who is surface antigen positive needs to be checked for Delta antibody. Delta antibody is about 95% sensitive. It's not perfect. You might have to resort to checking a Delta RNA by PCR. Now remember, Delta testing is available through Quest. It's also available through ARA. And I'm hoping that LabCorp, after a number of recent phone calls, conference calls I've had with them, will have, LabCorp will have Delta antibody and Delta RNA testing, maybe using even the diasorin kit, which is very, very high quality. So now the virus enters the cell. There's an uncoding process that takes place. And the nuclear material, the genetic material of hepatitis B enters the nucleus and forms this nuclear cccDNA. Now, cccDNA has at least 21 different morphologies, but this is the template for then making RNA, which could be full-length RNA or could be subsegmental RNA sequences that then code for various proteins. So you have pre-S1, pre-S2, S. We have other genetic material like the X protein, the polymerase. There's binding that takes place. You have core protein. You've got subsegments of the core protein, including the E-antigen. So this RNA is very, very important. And we're going to be talking a lot today about this RNA interference that's going to be, I think, one of the cornerstones of our therapy in less than 5 years, iRNA or RNAi. RNA destabilizers, I won't be talking about much today. Arbutus did have one in development. That development was stopped. It's not exactly clear why, but it may have been stability issues or administration or off target, because you're messing with RNA, you may be messing with cellular or changing cellular RNA. So that's cornerstone one. I want you to keep about iRNA. Cornerstone two of the next wave of hepatitis B, I think, will be these CAM or these capsid inhibitors, also called CPAMs, which there's 2 different types. Not super important about the types yet, but those are moving forward with 2nd and 3rd generation. The number of companies who have these CPAMs, one of the leaders, of course, is another Bay Area company called Assembly. John McHutchison is the CEO of that company and really doing, I think, a great job moving that through with Rich Colono. So foundation number 2, you can think of almost like a Greek temple here. You got your cornerstone iRNA, another cornerstone or corner pillar of that being CPAMs. A third is these nucleoside or nucleotide analogs, which we already have, right? We've got TAF, TDF, and entecavir, which are highly effective at suppressing HBV DNA. They don't have a big impact on correlated antigen, modest on HBV RNA. And these are new assays that you need to be very familiar with. Correlated antigen will be important and eventually, I think, commercially available, probably has an LDT. It's available through Fuji Rebio, a Japanese company, and it's being used in all of the clinical development. We're also quantitating RNA. And that's how we know the interaction with nucleoside or nucleotide analogs. Now let's move to the far right side of this slide. One of the 4th, or I should say the 4th pillar, big pillar of this, is modulating the immune system. I think it's going to take a 4-drug combination to get to my short-term goal or recommendation. And it's not saying mine, I'm talking now from a community perspective, of getting S loss at 40%. Now, when I say S loss, it means no S, no HBV DNA, and we would like to see surface antibody appear, although it doesn't appear to be required for true immune control. Because there's a lot going on. It's not just an antibody control. There's all these T cell responses, dendritic cells, NK cells that are all involved with this viral control. And many companies are involved in this space with new forms of interferon. We mentioned lambda interferon, PEG interferon, Pegasus, PEG intron are still on the market. We're hoping they'll continue to be produced, although there's a rumor that Roche may pull PEG alpha, their Pegasus from the market, from production by 2026. We hope that's not the case. but there may be new interferons. There's toll-like receptor agonists, TLR7 and maybe more TLR8, which Gilead is developing. Roche also has a TLR7. Now, RIG-I was one that was in development, but unfortunately it led to liver toxicity and even a patient death. So that RIG-I agonist that was in development has stopped. STING agonists, another immune modulator, are being looked at. And there's a variety of other medications, including checkpoint inhibitors, that may actually suppress hepatitis B. There was a nice paper from Ed Gain on combining that with a therapeutic vaccine with a nucleoside analog, and a couple of patients either had a marked decrease or cleared S-antigen by inhibiting these checkpoint blockade that's there. Insufficient B cell response, there are companies developing antibodies, bifunctional antibodies even, that can actually bind B and T cells and provide a better immune stimulation. Therapeutic vaccines, CAR T cell or T cell receptor engineering are also being looked at. So this is a very, very busy field. I've been very involved with different companies, including talking to investors, telling them, well, you saw what happened with hepatitis C, which was ignored for a long period of time from an investment perspective. And it changed the world for our patients. And it is investors that lay the foundation for these steps forward, even though there is progress in NIH and other basic science people globally. Okay, let's jump into some details. I want to recognize this wonderful organization called IHEP, and you'll see some of the other slides from different meetings, including EASL, where these slides are available for share. Okay, we're going to talk about siRNA. I'm not going to get into all the incredible complex details on this slide, but I'm going to have you focus on one thing. If you look across the title, there's this GalNAc. This is a way of delivering the siRNA to the hepatocytes. If you give siRNA intravenously, number one, it can be broken down. Number two, it can be delivered to a variety of different sites. And this GalNAc binds to a cell surface protein in the hepatocyte, allowing delivery. Now, this is a drug being developed by Arbutus, AB729. In the background, I want you to take a look at that top line where it says targeting a single X trigger. Now, by targeting the X trigger, the X RNA, taking that RNA into what's called the RISC complex, and then slicing that RNA, it also appears to affect all HBV transcripts. Now, I want to mention the Arrowhead product, which I'm not going to talk a lot about today, which is now being developed by Janssen as part of Johnson & Johnson. It targets two different parts of the HBV RNA, that genome, the pregenomic RNA, including the X region, and then on the opposite side of that genome, because it's basically a circular structure, binds to a second site. So by targeting two locations, having two triggers, there may be higher efficacy and there may be lower risk of resistance. Because anytime you have a drug that targets a viral protein or a viral intermediate like RNA, you do have a risk of resistance. And we actually already seen resistance to the capsid inhibitors, the CPAM inhibitors. You can see here a reduction in surface antigen levels on the lower left side out to week 24. And then really what we see a lot of times with the RNA is what looks like a reset. You give this for a short period of time, you have a reset where long-term, the S-antigen may be down a half a log or a log and a half. This is also important. On the right side graph, you're seeing reductions in HBV DNA levels across all these patients that were here. Now, there's a lot of complexities to this in terms of, are people coming in E negative or E positive? Are they coming in nuke naive, which is this study, or are they on nukes? And by looking at all these different combinations and using adaptive study design, we're hoping to be able to tease out what's going to be the best thing for these patients. I want to make a really important point, how heterogeneous the hepatitis B virus is with pre-core mutations, core mutations, and genotypes. This goes out A through J. Many genotypes. There's a lot of genetic variants, and then there's recombination that takes place with this virus in terms of the genetic materials. It's quite complicated. All right, so this is a single dose of GalNAc in the absence of nuke, and this is E negative patients. This is individual studies or individual patients now with their subject number. What I want to highlight here is reduction in the middle of HBV RNA. That's in the black mark. Blue line, blue circle, correlated antigen. Correlated antigen is an assay that looks at 3 different parts, a core, an E, and a P protein. But all of these really look, the correlated antigen looks like it's very closely monitoring what is happening at a transcriptional level from the cccDNA. It might even relate to how much cccDNA is in the liver. Why is ALT important? Well, we're trying not to see too many flares, although some people want low-level flares to say the immune system is being reconstituted. So this has got some broad-based antiviral activity, including S-antigen reduction, which is important, although not all drugs have an effect on S. Now, this is hepatitis B patients now, GalNAc AB729. And we're looking at the reduction and repeat dosing now of S-antigen and how many patients are even getting less than 100 IUs per ml. Now, why is 100 IUs per ml important? Well, first off, I make a message, all of your hepatitis B patients, you should have quantitative S-antigen in the chart baseline. And my recommendation is to check once a year. I want to put in a little advertisement for later this week to be on the meeting that I'm co-chairing with Harry Janssen for AASLD. It's a webinar that's specifically on quantitative hepatitis B surface antigen, how to use that in practice, and also how we're using it in drug development to some level. The quant S here, and a lot of these patients are below 100, and when you get below 100, that's a really important marker for a good chance of clearing S antigen. Very few adverse events that took place here. So very safe. Again, non-cirrhotic patients being looked at. All right, so a little bit more on correlated antigen. If you look at the left side, I want to make a comment on correlated antigen. There are some limits to this. It's got a relatively narrow range. It's not very sensitive, but we are seeing a half log decline approximately in some of these patients also. And then we've got this S-antigen response, but the different cohorts look quite similar. So it looks like we might be able to dose this subcutaneously every 2 months, which would be amazing because we're really trying to stay away from injectables as much as possible for these patients. Now, this final slide for this group is looking at, actually there's 2 slides here, just talking about cytokine changes. So we want to be seeing, as we suppress virus, changes in immune response. This will give us hope that as a single drug or 2 drugs or 3 drugs, there may be immune reconstitution that may obviate the need for having an additional immune modulator. I consider these immune signals to be pretty weak. I'm saying there is something happening here. I still think that 4th pillar of immune modulation will need to take place. This is really looking more at a granular level, looking at S reduction and looking at HBV specific T cell activation and looking at that dynamic. The hint here is that yes, you're dropping viral proteins, viral intermediates, and yes, you're seeing some correlate with immune reconstitution, immune reactivation. So there is some restoration of HBV-specific immune response. Now I'm saying it's not complete restoration, but we're seeing some signals that are there. Everybody who's in this space really thinks you need to have an active immune system. But the controversy here is if somebody moves from age 20 with hepatitis B, let's say they acquired it at birth, to age 50, there's this immune exhaustion that takes place. I don't like the term too much, but it's pretty standard. This immune exhaustion might even result in clonal deletion of different immune cells, including T cells and B cells. Clonal deletion may mean that in older patients, immune modulators won't have any effect. We'll just have to use suppressive therapy in those individuals. So more to come on that, but this is a dynamic. And I highlighted the complexities of the virus. Now we have a complex immune system. It goes from this immune tolerant, another term I don't like, but we use it, to immune active, to immune clearance, immune exhaustion, and then clonal deletion. Okay, let's now talk about this VIR-2218, another iRNA, again, targeting the X gene. Now, the X gene is this important transactivator. It may be involved not just in viral replication control, but also in oncogenesis that's here. This is also GalNAc conjugated. You can see all these different therapies, and then using this with pegylated interferon. In the upper right corner is a nice little diagram of how complex that hepatitis B virus is. You can see there that if you target the X protein, which is the orange line on the bottom, you have a trigger that cleaves that part of the RNA. You're really cleaving most of the other segments of the RNA, although it's not clear that part of the RNA that codes for S-antigen is being cleaved. The Arrowhead product, which I call it Arrowhead, although it's now part of Janssen or Janssen, actually has a trigger that goes on the opposite side of where the X RNA comes off and also cleaves that S RNA. This is on treatment data, phase 2 study. This is using iRNA in combination with PEG alpha 2A. That's Pegasus that's here. And again, a lot of details here, safe in the sense that you saw some side effects that were most likely interferon related, modest changes in ALT, although one patient got up in the 250 range, but S-antigen reduction that looked best when you're using the iRNA in combination with pegylated interferon. So pegylated interferon may be like our ribavirin world. It's going to be around for a little bit longer meaning in the 3 to 5 year time period. Minor adverse events, S log decreased by at least, S reduction by at least a log at week 12. So if you don't get this log signal of S reduction, you need to go to a higher dose or go to an alternate target in the iRNA world. When we talk about CPAMs, I just want to highlight, you don't see much change with CPAMs in terms of S reduction because they're working on a different mechanism, which we will get to shortly. Okay, so we talked about iRNAs. These are molecules that bind RNA, but we have another way of binding RNA, which are these antisense oligonucleotides. They're structurally set up that they're stable, they can be injected. They also are using GalNAc and other methods to get to hepatocytes, but the GSK product, which a lot of you know Terry Wright from the Bay Area, she's working with GSK on this development. They actually can deliver better and get better response, not using a liver targeting for some reason, but that's still to be teased out. So they had this phase 2A study and just looked across this period. They had injections that were 6 over about a 1 month period, and then 4 week period, and then follow up for 26 weeks. You can see in the central graph at the bottom, this really nice S reduction graphically. And then you can see on the left side, more information on the S and the log change at the different doses. There were some ALT changes here. There is data that these ALT changes were related to immune reactivation, immune reconstitution, not drug toxicity. So in many of these studies, there's a fear of ALT changes, but there's also a hope that you see an ALT change because of its reflection on immune system. A very, very complex graph. I'm not going to go into huge detail, but this is the level of sophistication that's going on in drug development. Along the left side, they're looking at acute phase protein. They're looking at liver enzymes, not liver function, collagen, innate immune response, leukocyte responses, different inflammatory and anti-inflammatory coding and responses. So it really was quite interesting that this ASO also targeting and binding HBV RNA, working through a different mechanism in terms of RNA breakdown and the risk complex, but a lot of immune reactivation that's taking place here. So I think there is a lot of hope, and maybe this will obviate that fourth pillar that we've talked about if we can get this level of immune reactivation. All right, let's move on into the next world, which is capsid assembly modulators. I'm going to speed up just a little bit because we want to have time for, of course, for Q&A here. So capsid assembly modulators block those capsids, that house that's made up of core proteins specifically, from forming, or even though it may form, it will be empty. It won't bind the HBV RNA, which then binds inside the capsid and then makes the HBV DNA and then leads to release. So they had this very profound, very good study, double blind, that open label out to 76 plus weeks. They had virologically suppressed E negative and E positive patients Then they looked at an off-treatment analysis that took place. The bottom line here is that all of the patients who had suppressed HBV DNA and RNA ended up relapsing. So this looks like it's either going to be a chronic therapy or they need to go to their second and third generation products that are here. So these patients were suppressed, for some profoundly suppressed, and when you took them off therapy, HBV DNA rose to substantially higher levels. RNA also rose. As I mentioned, there weren't much changes in surface antigen. Correlated antigen was also suppressed, but also increased, and the correlated antigen, the lower level of the correlated antigen predicted what happened after stopping therapy in terms of relapse and where that person had a reset. So the correlated antigen ended up being quite useful here. Really very few safety signals here. There were a few patients, of course, who had flares in the off-treatment period and then in the restart period. So monitoring these patients closely and retreating them. There were no patients with liver failure. There were no deaths. No S clearance from this, and whether S can actually be cleared from the CPAMs is not clear. I think they're going to be more like, I'm going to call it a sofosbuvir. There'll be a pillar of this, but they're going to require a number of other mechanisms to end up resulting in viral control. The CPAMs also appear to have an effect on decreasing cecDNA formation. All right, we're going to switch now to NAPs, nucleic acid polymers. These are oligonucleotides that are sequence independent. These are different bases here, charged and uncharged. They form this amphipathic alpha helix, and they block release of viral particles, including all of these different viruses that are listed at the bottom of here. I don't know if they have any information on coronavirus, but they've got a lot of other, our coronavirus, but they have information in general on coronaviruses. So there is this idea about the subviral particles having filaments and spheres, not being able to fully form, therefore not being able to package hepatitis B, grabbing the core that's got the HBV DNA and releasing that. So by inhibiting this translocation, by inhibiting this packaging, we decrease viral release, and we can decrease circulating S-antigen by 3 or 4 logs. This antigen is stabilized through specific cellular techniques where this NAP appears to disrupt this. And there's a lot of interesting information that came out at EASL on this from another company in the Bay Area called Aligos, where Larry Blatt is the leader, looking at how this works and interacts with heat shock protein and different chaperones that are there. They've worked on optimizing this. It's still intravenous, but they're working on a subcutaneous form. But on here, you can see these profound reductions in surface antigen when you look at the orange lines, which is tenofovir, TDF, PEG interferon, and these NAPs. And at the bottom, you're seeing antibody response. So this is that immune reconstitution that we talked about. When you clear S-antigen with this combination, you're getting an antibody response. It looks like it may result in better immune control. This will be combination therapy. You got TDF, you got PEG interferon, you've got a NAP. So it goes back to my 4 pillars. You got at least 3 pillars that are here. And this is looking at ALT. This is the scary part of this medication, in my opinion. You're having ALTs in the 400, 600, 800 range. They didn't have any liver failure. They haven't they haven't demonstrated any changes in bilirubin. But I think if you get this into a community setting and you're not identifying safely patients with cirrhosis or liver dysfunction, this may result in flares that could result in liver decompensation. But it looks to be at this time reasonably safe, taking into account these transaminase flares. So a second generation of the NAPs is something that I mentioned a few moments ago from Oligos called STOPST, S-antigen transport-inhibiting oligonucleotide polymers. And this looks at mechanism of action. This is in vitro information about intracellular S-antigen and inhibiting the HBV polymerase, how much extracellular S-antigen you see with marked reduction, decreases in E antigen, decreases in HBV RNA and core. So this Aligos product is going into human trials. There's a little bit more of the basic science. It doesn't appear to colocalize with surface antigen. It also is not activating any of these nucleic acid sensing immune receptors, which could explain some of the ALT changes that we see with the NAPs. So this may be a better medication, but we're going to wait till we see what happens in humans in clinical trials. Upper right-hand corner, some effects on core, some effects on HBV RNA. They're very effective at reducing S-antigen release. By reducing S-antigen and enhancing proteasome degradation of S-antigen, of course, you decrease toxicity of S-antigen from accumulation, but this may allow immune reconstitution to take place, because I'm a strong believer that S-antigen is modulating the immune system directly. Safety looked quite good. There's a lot of data on PK. I'm not going to go into all this in detail, but just in the blue box at the bottom, it looks safe and well tolerated with single and 3 weekly doses. They're thinking about going up to 120 milligrams. This may have a better effect on surface antigen secretion. And first efficacy results in Hep B patients is what we're going to be waiting for. Well, we talked about antibodies and being involved in immune control. There's been a lot of research in the past trying to get antibodies to be administered intravenously to modulate hepatitis B. We haven't had good luck. And in fact, there was a company in the Bay Area in the 90s that had a monoclonal that was given to post-liver transplant patients. That monoclonal resulted in S-antigen mutations and escape, and patients died of fulminant liver failure from hepatitis B with fibrosing cholestatic hepatitis. So we have to be careful about antibody, but these antibodies being developed by Vir, it's 3434, is something that appears to potentially block viral entry. It may block presentation to T cells and have a vaccine-like effect, and maybe involving clearance of S-antigen by delivering S-antigen in immune complex to dendritic cells. Now, we always have to be worried about immune complexes because immune complexes, as you can see with cryoglobulinemia, can be damaging to a variety of different organ systems. But on the other hand, these immune complex, by modulating immune complex and presentation, you might be able to ramp up and generate effector T cells. So there's this healthy volunteer, virally suppressed with low S-antigen, and then virally suppressed with any S-antigen. I want to talk about another complexity here of these trials is the issue of how high or how low the S-antigen level is on entry. There's a study that we're recruiting for in San Jose through the practice San Jose GI, which is one of the practices where I'm a partner. And that Altimmune is a therapeutic vaccine, and we want patients there who have low S-antigen levels. So if you want to help us recruit patients for clinical trials, make sure you have S-antigen in the chart for these patients, because those low S-antigen patients are probably most likely going to be the best to help. Now, this is an interesting slide in the sense of the distribution of patients. We've got Asian, Caucasian, and Black patients in these studies. So it's really important to have this diversity. In my clinic in San Diego, we have a large number of patients from East Africa and Sub-Saharan Africa that we see in clinic every week with hepatitis B. So this really is a global disease that we see, and as well as adult-acquired and high-risk individuals. So look what happened in S-antigen with this monoclonal antibody, S-antigen reduction by that magic 1 to 1.5 log. And on the right side, we're not seeing any ALT changes. So there's no, looks like significant immune modulation that would be a concern. All right, we're getting close to the end here. This is therapeutic vaccines. This is a VBI, it's, or now also called BRII-179. This also just talks about T and B cell response. Therapeutic vaccines are being focused on core and S. There actually was a core vaccine for hepatitis B that was being looked at in the 1980s, and the core vaccine looked as good as the S vaccine. But they're looking at non-structural proteins, such as the polymerase also, that can bring about an immune response. And maybe you're going to want 2 or 3 of these different proteins together to bring about this immune reconstitution. So these dose levels with and without interferon appeared to be safe and well tolerated. There was an induction of an anti-S response in more than 30% of these patients. Of course, you're not going to want to just see antibody response, but you're going to want to see reduction an S-antigen or some of these other important intermediates such as RNA, correlated antigen, they're E-positive, E-antigen. But at least they did see an immune response that took place here, and this is going to go into further trials, including combination therapy. I'm going to wrap up with a very important study. There were two studies like this. One actually are shown preliminary at AASLD, more data here at EASL. So actually looked here at 90 patients with chronic hepatitis B, 40 patients, surface antigen loss, in a treatment naive control group with chronic hepatitis B. Then they looked at intrahepatic cecDNA, and they're also looking at integration. And they did deep whole genome sequencing, looking for the number of integrants and the integration site. In the integration analysis on the right, small number of patients, 7 controls, 3 with functional cure. I'm just going to summarize this slide to say that the people with functional cure had much, much lower number and sites of integration. Integration is a pre-cancer event. This is what we think brings about carcinogenesis, of course, in combination with inflammation, a variety of other cell signaling that takes place. But if you can decrease cecDNA, that decreases the risk of reactivation, decreases the probability of relapse. And if you decrease the number of patients with integration, we're projecting that those individuals will have a much lower risk of cancer. And in our world, where we're suppressing most of our Hep B patients that are in practice, especially people with advanced disease, it's not transplant or liver failure that the patients or we are worried about, it's liver cancer. So I think we're going to be moving as this data matures to treating earlier and earlier, before patients get this clonal deletion, before they get immune exhaustion, and try to treat early to decrease the number of integrations or decrease the trajectory for the number of integrants. So in conclusion, I've got ABCD here. I want to just wrap up with some very, very important messages, which have to do with testing all adults for hepatitis B. And what really my message is here, a new initiative that we're trying to bring forth is everybody should know their ABCs. And of course, you have hepatitis A because we have epidemics. You have hepatitis B because we have great treatments. We have very effective vaccines. We have hepatitis C, test, treat, and cure. We have the coronavirus, right? That's the other C, know your ABCs. People are coming in for coronavirus testing. They can be educated about viral hepatitis testing, maybe even linked to testing. And of course, Delta. Delta, D is for deadly, right? We have new therapies evolving, but treatment isn't just giving a pill or an injection. It's about Delta, finding other family members or contacts who may have Delta, monitoring the Delta patients more closely for cancer, for progression. There's lots you can do for a Delta patient, even before we're going to be talking about administering some of these new drugs that we're hoping that will become available. Really want to thank our Northern California, Southern California colleagues, Stephanie for your kind introduction, Marina for supporting this so well, Robert for your great leadership here, and all the people at PaceMed for bringing this forward. Really want to advertise or advocate that we get a Delta talk on the schedule for next year, because we're close to having a therapy approved. And we really need to test all hepatitis B patients for Delta. And with that, I'll turn it back over to the sponsors. I'll stop the screen sharing and let's chat.
Okay, thanks so much, Dr. Gish, for your really informative and very thorough talk. We do have some questions in the chat, so I'll start with the first one. So since Hep B is sexually transmitted and it is a carcinogenic virus, why don't we treat all patients with detectable viremia?
Well, thank you very much for that, Stephanie. I see that came from Michael, a colleague in Fresno, who's on the front line. He's an advanced practice individual. So that's really where we're going, Michael. I think in 3 to 5 years, once people are found to be surface antigen, they get a DNA, the DNA is positive, we're going to start treating those. But we really need to have a hope for better S loss. 10% is the best we're seeing in E-positive patients. The negative patients, we might be able to get to 10% by doing what we call stop protocols, right? You got people on therapy for 3 to 5 years, they're DNA negative for 2 to 3 to 4 years, and then they have a low surface antigen. You stop therapy, make clear. But 40% is sort of a magical number where we're going to cross another threshold where we can engage people in more complex treatment. But that's right on the mark, exactly my first and last point of my talk. Thank you.
Our next question, is there any data for the practice at many centers to give Hep B vaccine boosters in immunocompromised, for example, IB patients on anti-TNFs, based on an anti-Hep B surface antibody positive titer?
The answer is,
what is your practice?
No, there is no data. Boosting came about because it made us feel better. Oh, we gave a booster, we got more surface antibody, but no data. And there was a study from Japan that I saw in abstract form. I haven't seen the final publication, where they went into immune suppressed patients, chemotherapy, cancer clinics, boosted their surface antibody to try to prevent reactivation. That's the other setting, which are core positive. Vaccine had no effect on reactivation risk. So boosting, there is no data. The guidelines talk about boosting HIV patients, dialysis patients, and then a couple other high-risk patients. There is no data to say this is decreasing transmission or decreasing reactivation. So we really should be evidence-based here.
Okay. And then any advice for counseling with positive Hep B virus patients that have a low viral load who wish to be sexually active with a partner? What is the infectivity risk for the other partner?
If the partner is vaccinated, no risk. And it is just give 3 doses and you're done. But of course, before you give the 3 doses, make sure you've got a panel. The vaccines are phenomenally effective. And remember, these aren't just B-cell vaccines, getting an antibody response. All of our vaccines, including HBeAg-B, which is the best of all of our vaccines available globally right now, has a profound T-cell effect. And T-cells ultimately also help control or protect us from getting infected.
And then a quick follow-up question to that is, if one partner is Hep B positive and the other is Hep B negative, any advice for if they're considering conception?
Yeah, that's a great question. And I think it was maybe Kevin Mann who sent me an email about this last week. In California, there was a state law that said if you have an infectious disease, you can't be involved with any type of fertility treatment. But if the partner is vaccinated, there's no risk of transmission. There's no risk of that partner getting hepatitis B. Hepatitis B vaccine is fantastic. It's as good as our Pfizer or as our Moderna for coronavirus. They're really great. And you might see occasional patient, and this is if you look over 20 or 30 years, you can see occasional patients who got vaccinated, lost surface antibody, and then became core antibody positive, but surface antigen negative. That means the vaccine worked. Yes, they got exposed. They had a tiny Hep B infection, cleared, controlled. Vaccines work. We vaccinate 3 times and say goodbye. We don't check titers. We don't do follow-ups on those people.
Okay. Do you have any more information about the approved drug for Hep D?
Great. So the approved drug is bulevirtide. This is at the super beginning on that little diagram. We were talking about an entry inhibitor. It's approved in 3 countries in Europe, which I think is France, Belgium, and Austria. Conditional EMA approval. It's a chronic injection once a day, indefinite therapy, but it results in profound delta RNA control. There's also data about using this in combination with pegylated interferon and nukes, including TDF, and that could also result in maintained virologic response in 30 to 40% of patients after a year of therapy. So bulevirtide, there's a global study that's wrapping up. I'm hoping it will be approved by the end of next year by Gilead, available in the US, and they are going global with this. So you're going to be seeing a huge amount of delta information come out. We have many publications in the work and an awareness campaign.
Great. And then next question, what are your thoughts on the subset of potential patients that don't mount detectable surface antibody response after Hep B vaccine? Is there any data on boosters here, or do we assume after completion of the series that all are protected regardless of antibody levels?
I'm going to take a strong position here, Stephanie, and say you give 3 doses, don't check titers. Done. You're done with that patient. You look at the guidelines, the only places you might look at titers, again, I mentioned no evidence, but you look at titers, dialysis, HIV positive, especially if they're sexually active, and super high risk, say a vascular surgeon, or maybe a transplant surgeon who's bathed in blood all the time. Give your 2 doses of HBeAg, which is the best, or 3 doses if you're using Merck or GSK, and say, you're done, you're protected. This is a B cell vaccine, it's a T cell vaccine, and that patient's protected. And we think lifelong. We have data out over 20 to 30 years. Great study in Senegal, more than 20 years. Data in Alaska from Brian McMahon, incredible protection out to 30 plus years.
Okay, so just that follow up. So unlikely that people will lose immunity over time after 20 to 30 years then?
I'm saying they don't. They have memory B cells and memory T cells, and don't check titers. They're protected, and just tell people be safe, safe sex, avoid blood exposure.
Okay. Another question, do these promising ASO RNAi therapeutics have a mechanism to eliminate cccDNA, or will these be lifelong suppressive therapies like NUCs?
Fantastic question. So cccDNA is a dynamic. It's in the nucleus, but it requires continuous replenishment. So once the RNA goes out into the capsid, and the core attaches the core protein, forms a capsid. The capsid actually recycles the RNA back into the nucleus, gets reverse transcribed into DNA, and gets replenished. So if we can reduce capsid, we can reduce RNA, we're going to reduce cccDNA. So I think that's already happening with some of the drugs. There's some hints, especially when you see correlated protein going down. It's definitely meaning less transcriptional activity of the cccDNA. And then there's this turnover that takes place. And the half-life of cccDNA is a big controversy. Some people propose 35 days, some people propose 140 days. But if we can reduce replenishment, we can potentially get down to a low level. I didn't mention another drug, which are called endonucleases. There's also a very famous set of compounds called CRISPRs, right? CRISPR-Cas9. If we can deliver these to the liver cells, we can cleave cccDNA and really enhance the depletion. But don't forget, we have integrants. This virus integrates. The integrants produce S-antigen, a truncated S-antigen, but they're producing S-antigen long-term, which may fool us. Just think, you get rid of everything in the cytoplasm, you get rid of all the cccDNA, but there's still a sprinkling of S-antigen coming off the integrants that are there. But the good news is the integrants are not full-length virus sequences. You're not going to make a full-length virus from the integration.
Okay, thank you. What is your approach to treatment to a newly diagnosed patient new to the country that is 28 weeks pregnant with an extremely elevated viral load?
Fantastic. So we're talking about viral load here for hepatitis B being over 200,000 IUs. That's the magic number where you cross a threshold of risk of transmission from mother to baby. The drug that we have the best and most data on is TDF. The drug we have the least information on is entecavir. So I would not use entecavir. I would consider TDF. But there's a great study that was released just recently with Calvin Pan as the senior author about using TAF to prevent vertical transmission. And it might even be a little bit better than TDF. It appears just as safe as TDF. But 200,000, get the mother on TDF or TAF now. The other great advance that came out of the Jourdain article from New England Journal that took place in Thailand is that if you give the hepatitis B vaccine within 2 hours of birth to the baby, we may not even need HBeAg. Now, of course, in the US, we're going to use HBeAg, But there's over 1,000 babies born in the US surface antigen positive every year. So we are failing somewhere. So nukes to the mom if over 200,000, baby gets vaccinated at 2 hours or less. And of course, we'll be using HBeAg for the near term. That could take our transmission rate to 0, in my opinion.
Great. Any other questions for Dr. Gish? Okay, well, thank you so much for your time. Thanks everyone to all their great questions as well.
Appreciate being invited. Delta next year.
Thank you.